Today we're going to finish looking at chapter 12 by looking at plant growth, hormones, and responses. We're going to focus first on meristematic tissue, which contains undifferentiated cells that can still perform mitosis. They are regulated by hormones, and we see hormone production and regulation also occur in animal cells, not just plant cells. Hormones are chemicals that circulate through multicellular organisms, usually through some type of vessel system. In animal cells, they tend to move in and out of interstitial fluid. But hormones regulate cellular processes, and they do so by interacting with receptors on target cells for that specific hormone. There are 5 identifiable groups of plant hormones: auxins, gibberellins, cytokinins, abscisic acid, and ethylene. And there's a group of fluorogens, but they haven't been 100% identified. But we do think they exist. Here we'll look at auxins first, and auxins in plant responses have to do with stimuli such as light, gravity, and touch. They help generate responses related to phototropism, gravitropism, and thigmotropism, which are all growth responses to things like light, gravity, and touch. And they are going to be examples of nastic movements, or movements that occur in a pre-programmed way, like when flowers close at night to prevent water evaporation. And the direction of the tropism is dependent on the direction of the stimulus. For example, with phototropism, we see that plants tend to move or grow towards light. The auxins accumulate in areas where we see increased cell elongation. In this case, we see auxins accumulate in areas where there is less light because we think that auxins are destroyed by light. So they end up in higher concentrations on areas that are further away, but those areas further away via elongation actually turn the stem or the shoot system towards light. So we see this growth imbalance that bends plant stems. There's also an extreme form of phototropism called edivolation, where if you place a plant in a dark area, none of its auxins are destroyed, so you see tons of shoot growth. They actually grow so quickly that the stem becomes very long and thin, and after a certain time, if that stem cannot find light, the plant will die. Gravitropism in shoot systems is seen when the stem grows upward even after being tilted. We can see here that the auxins accumulate towards the bottom, or due to gravity accumulate at the bottom of the bent stem, but that's going to be the area that experiences elongation. And we'll actually see the curve of the plant shoot system move upward. So this is going to be a shoot that opposes gravity or is acting negatively gravitropic. Root systems are actually positively gravitropic. So they're going to move downward with gravity. So stems grow upward, opposite gravity, roots grow downward. So their effects or responses to auxin accumulation are different. The roots, again, are going to be positively gravitropic. They'll go with gravity. And then negatively gravitropic is how we would describe stem growth, thanks to auxins. And in thigmotropism, we can best illustrate with a vine. Vines tend to wrap around whatever they touch, and the leaves of the vine always point away from the structure upon which it is growing, and that's due to thigmotropism. Auxins, in this case, tend to move away from the surfaces that a plant touches, elongating the sides that are not touching a surface or substrate. And that's why stems and leaves grow or continue to wrap around a particular structure. And the vines are generally seen, and they can be seen here actually in the right, touching and growing around in a very predictable pattern. So growth will be inhibited on the inside, the part of the stem being touched. So cells elongate on the outside, the part not being touched, and that's due to auxin accumulation on the outside or on areas that are not touching a structural support. And that's going to cause the stem, in the case of vines, to coil. Let's look at the next group of hormones called cytokinins. They produce effects opposite to those of auxins, so they can affect mitosis rates by stimulating cell division. They can stimulate cellular differentiation. They can cause cells to grow thicker, not necessarily longer, they actually inhibit cell elongation, but they do allow different tissues to grow wider. They even allow leaves to expand. They affect the synthesis of chlorophyll. They're produced in multiple areas of the plant: roots, embryos, and fruit. And while auxins were mainly stimulating parts of the apical meristem to stimulate mitosis and growth via elongation, they mainly made the plants grow taller, essentially. Instead, cytokinins are going to stimulate the lateral buds. The lateral buds can still be considered meristematic tissue, but they're not at the top. They're not the apical meristem. Instead, they're at the sides and are responsible for branching, so it makes the plant grow fuller, not taller. Here we can see apical dominance compared to bud decapitation that eliminates apical dominance and is a result of pruning. So auxins tend to act on the apical meristem at the top of a plant where we see tissue that can perform mitosis, and new tissue being generated here would make the plant grow taller. And the terminal bud there at the apical meristem releases auxins, and that's why we can say that auxins are related to making a plant taller or related to that type of growth. And while a plant is growing along the terminal bud at the apical meristem, we see an inhibition of growth of the lateral buds, or a decreased amount of growth there. But if you actually prune or remove the apical meristem, you can inhibit growth in that we're going to keep it from becoming taller, but we will now see most of the growth at the lateral buds. And these buds, due to the selective pruning of the apical meristem, are now going to no longer be as dormant, and they'll start to develop and produce branches of their own, which creates a bushier plant. The next group is the gibberellins, and this is a family of hormones that promotes elongation of stems and leaves, but it has other jobs as well. So some of its jobs are similar to the auxins, but mainly it's going to focus at the tips of stems and roots and less at the apical meristem. It also focuses on fruit. It causes it to grow larger. Growers of seedless grapes, for example, usually would apply gibberellins to make the grapes grow larger and the stems longer. Flowers get sprayed with gibberellins so that they're bigger, their blooms are fuller. And gibberellins are also used to increase the yield of tomatoes and berry crops. So here we can see the application of gibberellins and how it has produced a larger size in the fruit, specifically of grapes. So active gibberellins have a lot of physiological effects, especially on the fruit, which is protective of embryos. We can also look at the abscisic acid group, and ABA or abscisic acid affects the shoot of plants by causing stomatal closure, reduction of leaf mesophyll, reduction of leaf conductance, induction of leaf senescence. It can cause leaves essentially to fall off, control of leaf expansion, cuticle wax formation on leaf surfaces, and inhibition of shoot growth. In the root, it causes root elongation, inhibits lateral root formation, and increases root hydraulic conductivity. But I want you guys to focus specifically on how ABA's primary function is to inhibit the abscission layer so that it doesn't close off. So it's produced mainly when we have mature green leaves, stems, and fleshy fruits, and then it's actually inhibited during the winter in conditions where we want to see the leaf fall off, because abscisic acid, when it stops being created, actually can lead to the desiccation of the abscisic layer, which can lead to the petioles of leaves falling off in the fall. And then ethylene, which we know probably the most, or if you've heard of it before, you know it the most, as a fruit ripening agent. And it's a gas essentially that promotes the ripening of fruits. It's actually used by farmers to artificially ripen fruits if they're picked before they should have been or before they were ripe. And ethylene also causes the abscission layer to close off. So not only the absence of ABA, but the addition of ethylene can cause petioles to break off of their shoots. And auxin concentration tends to decrease when ethylene concentration increases in the winter, and that is also going to initiate leaf drop. And without leaves, transpiration stops. That's going to help the plant retain water, and that is why it's occurring during the winter. And then briefly we'll look at phlorogen, which botanists aren't sure they've identified correctly yet, but they know something controls the flowering of an angiosperm. And they did recently discover a protein called flowering locus T, abbreviated FT, and that's believed to be the protein that's governing flowering in angiosperms. We also know phytochromes tend to play a role, so it might be kind of a multidimensional mechanism. And so we're just calling kind of whatever allows for plant flowering regulation to occur phlorogen for the time being. Next, we can look at some unique plant designs. So we can look at how they develop in certain habitats, as well as just some types of adaptations. For example, rice plants have air spaces in them so that they can live in water. Water lilies also have air spaces, and their roots have these air spaces all the way up to their long petioles, and that way oxygen from the surface can diffuse down through the underwater stem and into the roots. You have mangrove trees, which have modified roots that are made to take in salt, and that way the mangrove trees can live in salty marsh habitats. But they are not only just tolerant to the salt concentrations, they can actually eject this salt from their shoots and their root systems, and it ends up on the leaf surface, and wind and rain wash away the excess salt. Also, desert plants are made to conserve water. They can live in hot, very dry habitats because they prevent water loss. They're going to prevent water loss through the stomata by slowing transpiration at night, by closing their stomata. They tend to have less broad leaves that aren't flat. They tend to have things like needles that are very thin and spine like. And the spines themselves will lack guard cells and lack stomata. So their green stems essentially are what carry out photosynthesis in that case. The stems of cactuses are the areas that will swell during rainy periods to store excess water. That way, they have water for dry spells. And the spines can also be protective. They protect them from animals that are trying to eat them as a readily available food source. Now, because we have desert plants that prevent water loss by closing their stomata, usually during the day, sometimes also throughout the night, we have to acknowledge a modification of the C4 pathway. In this case, we are seeing CAM photosynthesis, which is very complex, but essentially we can boil it down to the fact that carbon dioxide can be stored at night when stomata are usually open as opposed to in the daytime when they're closed. And so that stored carbon dioxide can be used then during the day to carry out photosynthesis, even though we are not getting a rapid influx of carbon dioxide at the time that photosynthesis is occurring. And this storage of carbon dioxide before its fixation is actually going to be very different than what we see occur in the C4 pathway or C3 pathway. All right, we also have plants that have modified leaves, like these carnivorous plants. They generally eat insects, and their closure is triggered by usually some type of nastic movement of small hairs within the carnivorous plant. And the leaf would always constrict once stimulated, never open once stimulated, and it would actually produce digestive juices to decompose the insect it eats. Now, they don't use the insect for food, instead, they use the breakdown products for the insect to carry out their photosynthesis. So, they are going to use photosynthesis to produce their own food, just like other plants, and the insect is going to be used for biological biosynthesis or the generation of other chemicals. They're getting their raw materials that they need for biosynthesis from the insect they catch, but they're not getting their food. They still perform photosynthesis. And that's it for chapter 12. So next we're going to move into and look at chapter 13.